Good morning. So today we will discuss biosynthesis of fatty acids, right? Biosynthesis of fatty acids, right? What is fatty acid synthesis? What is fatty acid synthesis? Synthesizing the fatty acids, right? When will it happen or undergo in the body? Suppose you are eating excess sugar, right? What is sugar? It is a carbohydrate, any form of sugar, right? You are eating it in excess, large quantities, right? Or you are taking proteins or amino acids in also excess, right? The body needs, normal needs will be met properly, right? After that, the excess amount of these carbohydrates and amino acids or proteins will be converted to fatty acids, right? Today we are doing biosynthesis of fatty acids, right? When will the body go for fatty acid synthesis, right? There will be two conditions, right? Either you are eating excess sugar or carbohydrates. Normally, the requirement of body will be met first. After that, extra will be diverted for fatty acid synthesis. In the similar manner, you are taking proteins, the body utilizing for muscle building and all the activities, right? But excess will be diverted for fatty acid synthesis, right? So today's topic is fatty acid synthesis, right? And tell me the organ first, where it will happen. Organ, liver, kidney, adipose tissue, and lactating mammary gland, right? Three organs: liver, kidney, adipose tissue, and lactating mammary gland. Now organ is clear. Now tell me organally, or cytosomal fraction or mitochondrial fraction. Cytosolic, cytosolic, cytosolic और cytosomal fraction में होगा, right? So I repeat, you are taking excess glucose, you are taking excess proteins. Normally they will do the functions of the body. After that, the excess will be diverted for this fatty acid synthesis, right? Now coming on to the organ, organs are liver, kidney, adipose tissue, and lactating mammary gland, right? And it happens in the cytosolic fractions of the cell, right? Now third thing is that what are the raw materials? Raw materials for fatty acid synthesis. Raw materials from which fatty acid can be synthesized, right? The first raw material is acetyl CoA, right? The second raw material is NADPH, right? And the third one is ADP. From these three, we can synthesize fatty acids, right? Acetyl CoA will provide carbon atoms, right? And this NADPH will provide reducing equivalents, and ATP will provide energy, right? So fatty acid synthesis, organs are liver, kidney, adipose tissue, and lactating mammary gland. Fraction of the cell is cytosolic fraction of the cell, right? And three raw materials are required. What is that? First is acetyl CoA. It will provide carbon atoms. NADPH will provide reducing equivalents, and ATP will provide energy, right? That's finished. Now first source is acetyl CoA. Now it will come from which part? Acetyl CoA. Anybody who wants to add the sources of acetyl CoA? Hope you remember we did glycolysis in the first classes, right? We started from glucose. What will form? Pyruvate, right? And this pyruvate was converted to acetyl CoA via pyruvate dehydrogenase complex. From here it enters the TCA cycle. We started from glycolysis from glucose, and then the product was pyruvate under aerobic condition and lactate was under anaerobic condition, right? But this pyruvate via pyruvate dehydrogenase complex can be converted to acetyl CoA, right? And this acetyl CoA is raw material for synthesis of fatty acids. Now you can correlate, right? The second is like first acetyl CoA, one source is from glycolysis. You get pyruvate. From pyruvate via pyruvate dehydrogenase you will get acetyl CoA. Any other sources of acetyl CoA? One is clear. Any other sources of acetyl CoA? We did beta-oxidation in the previous class during even chain fatty acids. What are the two products? Acetyl CoA and acyl CoA, right? And acetyl CoA will also come from beta-oxidation. First source is pyruvate dehydrogenase complex via glucose, right, to form acetyl CoA, right? Second is our beta-oxidation of fatty acids, we will get acetyl CoA, right? Third source कौन सा है? From ketone body metabolism, we can also get this acetyl CoA, right? Hope you remember ketogenesis, acetyl CoA plus acetyl CoA, acetoacetyl CoA, then HMG CoA, and then finally the ketone bodies were formed, right? So there are three sources of acetyl CoA. One is from glucose, you will form pyruvate via glycolysis. Pyruvate is converted to acetyl CoA via pyruvate dehydrogenase complex. From here it can answer TCA cycle as well. But this acetyl CoA can also be used for synthesis of fatty acids. Clear? Second source was beta-oxidation, from where you can get acetyl CoA, right? Third was ketone bodies, right? And ketogenic amino acids, they can lead to synthesis of also, right? So all these sources will lead to synthesis of our first building block for fatty acids is acetyl CoA, right? Clear to all of you? Second source is NADPH. Tell me the source of NADPH from carbohydrate metabolism. First is HMP shunt. घर पे पापा रहेंगे. Hope you remember the mnemonic, right? HMP shunt is the only pathway that lead to synthesis of NADPH. Any other sources of NADPH or enzyme? It can also be formed from isocitrate dehydrogenase reaction, right? And also from malic enzyme. So there are three sources. One is HMP shunt, second is isocitrate dehydrogenase, third is malic enzyme for the second building block. To form fatty acid, you need three products. One is acetyl CoA, second is NADPH, and third is our ATP. What are their functions? Acetyl CoA will provide carbon atoms, right? This NADPH will provide reducing equivalents, and this ATP will provide energy for the synthesis of fatty acids, right? And acetyl CoA is coming from three sources. One is beta-oxidation, and one is via pyruvate dehydrogenase complex, this reaction, and the third is from ketone bodies, and fourth is from ketogenic amino acids. Clear? NADPH is coming from HMP shunt, isocitrate dehydrogenase reaction, and this malic enzyme, right? And the third is ADP, that will provide energy for ATP for fatty acid synthesis, right? Now the fatty acid synthesis broadly has 3 steps, 1, 2, 3, right? First step is providing acetyl CoA and NADPH, right? And the second is conversion of acetyl CoA into malonyl CoA, right? And the third is fatty acid synthase complex reactions, right? For the synthesis of, I repeat, when you are taking extra glucose or extra fat or extra adipoprotein, right? The body uses it for normal functions, the extra is diverted for fatty acid synthesis, right? The fatty acid synthesis requires 3 raw materials or 3 products. One is acetyl CoA, second is NADPH, and third is our ATP, right? And acetyl CoA, you will get it from, first is beta-oxidation, ketone body metabolism, and pyruvate dehydrogenase reaction, right? So we'll get our first product, that is acetyl CoA. Second is NADPH, you will get it from HMP shunt, then isocitrate dehydrogenase reaction, and third is malic enzyme. Clear? The third is ATP, that will provide energy for the fatty acid synthesis. Now, there are 3 steps for fatty acid synthesis proper, right? The first is providing acetyl CoA and NADPH, right? And second is conversion of acetyl CoA into malonyl CoA, and third is reactions of fatty acid synthase complex proper, right? Now this acetyl CoA, the first product, where is fatty acid synthesis going on in the body? Organ is liver and cytosolic fraction of the cell, right? But this acetyl CoA is in mitochondria. So you have to bring this acetyl CoA from mitochondria into the cytosol, right? So that it can form fatty acids, right? Now we have 2 compartments. This is our mitochondrial matrix, and this is our cytosol, right? This is mitochondria, this is cytosol, and this acetyl CoA is formed in which compartment? Matrix, right? But fatty acid synthesis is going in cytosol, right? So this acetyl CoA is not available in the cytosol for fatty acid synthesis. It is produced in the matrix. First process is we have to bring this acetyl CoA into the cytosolic fraction of the cell for fatty acid synthesis, right? How will you do it? We discussed 3 sources of acetyl CoA. One was from glucose to form pyruvate, pyruvate to pyruvate dehydrogenase complex, and then will form acetyl CoA, right? So normally we have, this is cytosol and this is matrix. Glucose is there. Glucose is converted to pyruvate. Tell me the reactions in which glucose is converted to pyruvate. Glycolysis is there. This pyruvate will enter here. Pyruvate can enter the matrix, right? Pyruvate will form this acetyl CoA via pyruvate dehydrogenase reaction, right? We discussed this reaction, right? And pyruvate via pyruvate carboxylase, it will form oxaloacetate. Oxaloacetate is the second last step of TC, hope you remember, right? We have pyruvate, which comes from glucose via glycolysis, right? And this is pyruvate. Pyruvate via pyruvate dehydrogenase reaction will form acetyl CoA and oxaloacetate, right? And this oxaloacetate and acetyl CoA will combine in the presence of citrate synthase to form citrate, right? And this citrate is permeable from mitochondria into the cytosol, right? We have citrate now in the cytosolic fraction of cell. This citrate via citrate lyase is converted again back into oxaloacetate. I will show you the reaction. That will make more easy for you. So, right, the three steps are there. First is production of acetyl CoA and NADPH, conversion of acetyl CoA into malonyl CoA, and the third is fatty acid synthesis complex proper, right? Now, see this reaction first. See, glucose is there, it is converted to pyruvate via glycolysis, and this pyruvate can enter the matrix. See, pyruvate via pyruvate dehydrogenase complex will form acetyl CoA, and pyruvate carboxylase will form oxaloacetate. These two will combine in the presence of enzyme citrate synthase to form citrate. This citrate is permeable into the here blue one. Citrate is coming out. Citrate is converted again back into oxaloacetate, then malate, and malate will finally form pyruvate, and pyruvate can again re-enter, right? And during this reaction, NADPH is formed. Our first raw material NADPH, and acetyl CoA कहाँ बना? See it during this reaction. See, right? This acetyl CoA was not permeable from the mitochondria to the cytosol. Now this acetyl CoA is available, the first raw material. NADPH is available and ATP are also there. Now we will start with the first step. For fatty acid synthesis, the three steps are there. First is availability of acetyl CoA and NADPH in the cytosol. Second step is conversion of acetyl CoA into malonyl CoA, right? So, anyone who has doubts in this reaction? Anyone? Okay, I repeat. Fatty synthesis takes place in which organ? Liver, kidney, adipose tissue, and lactating mammary gland, right? Organ is liver, organally is cytosol, right? And for the synthesis, you need three raw materials. One is acetyl CoA, second is your NADPH, and third is your ATP. ATP will provide energy, NADPH will provide reducing fuel. NADPH कहाँ से आएगा? HMP shunt से और isocitrate dehydrogenase reaction से, और एक और था malic enzyme वाले reaction से, right? And first was acetyl CoA. Acetyl CoA कहाँ से बनेगा? Glucose से via glycolysis, and we'll get pyruvate. Pyruvate via pyruvate dehydrogenase complex will form acetyl CoA, right? But this acetyl CoA is not permeable from the matrix into the cytosol. For this, we are using this reaction. Acetyl CoA with oxaloacetate will, in the presence of citrate synthase, will form citrate. Citrate will permeate out, and from this reaction we will again form oxaloacetate, and here acetyl CoA is formed. Now acetyl CoA from here is brought into the cytosol, right? Now it is in the cytosol, so it is available for fatty acid synthesis, right? We have all the raw materials, NADPH is there, acetyl CoA is there, ATP is there. Now we start with fatty acid synthesis, right? It is going in the which compartment? Cytosolic fraction of the cell. Organ is liver, kidney, adipose tissue and lactating membrane. When the fatty acid synthesis goes in the body, when we have extra calories or you are taking extra glucose, normally used for processes of the body, extra proteins, normally they are used for bodybuilding, everything, but extra is going for diverted for fatty acid synthesis, right? For fatty acid synthesis, you require 3 raw materials: acetyl CoA, NADPH, and ATP. NADPH comes from HMP shunt. Acetyl CoA formed in the matrix, but it has to be brought into the cytosol. Third is ATP, which will provide energy, right? Now we have all these raw materials in the cytosol. Now coming on to the second reaction, conversion of acetyl CoA into the malonyl CoA. Let's see this reaction. We have acetyl CoA. It is converted into malonyl CoA, and it is a carboxylation reaction. So what we will add? Carbon dioxide. Enzyme is acetyl CoA carboxylase, and ATP is formed during this, used in during reaction. ATP is converted to ADP and phosphate, and this reaction requires biotin. One thing, right? So we have now malonyl CoA. Clear? Anybody who has doubts? Active ho ja, back proper, yes, right? So we started from 3 raw materials: acetyl CoA, NADPH and ATP. acetyl CoA कहाँ से आया था? ग्लाइकोलिसिस से. ग्लाइकोलिसिस से क्या बनता था? ग्लूकोज से. पायरुवेट से पायरुवेट डिहाइड्रोजेनेज यूज करोगे तो बन जाएगा acetyl CoA, right? First raw material है, but this acetyl CoA is in the matrix and it has to be brought into the cytosol for the reaction of fatty acid synthesis. So it is brought via the previous steps, and NADPH comes from HMP shunt. ATP is already there, and now the second step was conversion of acetyl CoA into malonyl CoA. Acetyl CoA is there, and one more carbon is added. You can see it is added via carboxylation reaction. Enzyme is acetyl CoA carboxylase, and you need biotin for this reaction. Clear to all of you? The first 2 steps. Now we'll move to the third step. What is third step? Reactions of fatty acid synthetase complex, right? And this fatty acid synthetase complex is very important, right? It is very difficult to remember the 7 enzymes of the fatty acid synthase complex, but we will make mnemonic out of it so you can remember it, right? And we will also do it in the 3D, right? So for these reactions, you first need fatty acid synthetase complex. थोड़ा पानी दे दे वापस. Yeah. So just see the complex first. I told NADPH generators, one was HMP shunt, second was isocitrate dehydrogenase, and third is malic enzyme, right? Now this is, just see the diagram first. It is very difficult diagram, and you will get it for 5 marks minimum. मेरे पेपर्स में आया था. Yes, ये आएगा 100% आएगा, लेकिन we'll try to make it as easy as possible, right? So there are 7 enzymes, 1, 2, 3, 4, 5, 6, 7, and 1 is acyl carrier protein, right? Just first do the features. First feature is that it is a multi-enzyme complex, right? And it is in the form of a dimer, and the dimer is only functionally active, right? And there are 7 enzymes and 1 acyl carrier protein. 7 enzymes and 1 acyl carrier protein, right? And I told it is a dimer, right? In monomer we have sequence of 7 enzymes and acyl carrier protein, and then it will come in antagonistic manner. One from this side and other from this side, so it will form complete dimer, right? Dimer is functionally active and all the enzymes are originated from a single gene, right? And the other metabolites, they don't interfere in this complex, right? So let's see the diagram and try to memorize it. मेनोमिक बना रहे इसका, बोलो 5 मार्क्स का क्वेश्चन कवर हो जाएगा. फर्स्ट एंजाइम इज कीटोसाइल, सेकंड इज एसिटाइल ट्रांसएसाइलेज, थर्ड इज malonyl ट्रांसएसाइलेज, fourth is dehydrogenase, fifth is enoyl, no noise at the back, reductase, sixth is ketosyl reductase, seventh I told acyl carrier protein is attached, and seventh is our thioacidase. So these are the 7 enzymes. These are the 7 enzymes, right? So we use the monomic Ks, At, Mt, DH, ENOR, and Kr and ACP and TH, right? अभी पूरा बनाने थे फिर फोटो खींच लियो, right? So the 7 enzymes are there, right? The 7 enzymes are there, 1 to 7, and on the other dimer they are from this side, like this, right? So how will you remember them? Movie is there, Kabir Singh, right? What was going with Kabir Singh? He was having acidity, right? He was having acidity and he is very uneasy, right? So he went to Mother Teresa hospital, where? At Delhi, right? Kabir Singh was feeling uneasy due to acidity, right? He was residing at Delhi, right? So he went to Mother Teresa hospital. हो गए 4 enzymes first, right? Kabir Singh at Mother Teresa hospital, New Delhi, because he was having acidity, right? So he was having acidity. The doctor gave him ENO reductase. ENO से उसको क्या मिली? Relief. Enol reductase बन गया, right? Then फिर क्या हुआ? बाद में सीसीटीवी फुटेज में आया कि वहां से इनो चोरी हो गए फार्मेसी शॉप से, राइट? इनो चोरी हुए तो क्या करोगे? क्या एंजाइम आया नेक्स्ट में? इनॉल रिडक्टेज हो गया, कीटो एसाइल, देन एसीपी एंड देन थायोएस्ट्रेज. जब इनो चोरी हुए तो क्या हुआ? They checked the CCTV footage, right? And they found that cats have stolen the ENO, right? The CCTV footage was checked for the pharmacy and the ENOs were stolen by cats, right? The cats were caught and was handed over to ACP, right? And ACP took them to Thane or police station, that is our thioestrase, right? So this story can correlate. कबीर सिंह वाज देयर. कबीर सिंह का नाम क्या है एग्जैक्ट? कीटोसाइल सिंथेटेस, राइट. कबीर सिंह he was having acidity, so he went to Mother Teresa Hospital, New Delhi, right. At for acetyl transacylase, MT for malonyl transacylase, Delhi for dehydrogenase, and the doctor gave him ENO for acidity and he got relief in all reductase, right. Later on he bought further ENOs, but he went to pharmacy shop, but though they were stolen, right. When they saw the CCTV footage, they found that they were stolen by kitties, cats. So keto is ketosyl reductase, right. So the cats were caught by the ACP of police and he took them to Thane or thioesterase. So this mnemonic I hope can work, right. So see the enzymes. Now listen to one thing, that it is antagonistic. Ketosyl synthetase is there, here ketosyl will come. Acetyl, acetyl, malonyl, malonyl, right. 1, 2, 3, 1, 2, 3. Dehydrogenase diagonally, non-reductase, ketosyl reductase and ACP, right. And at the end there is thioesterase, right. I told it is a dimer, one monomer, one monomer. The dimer is functionally active, right. And it comes antagonistic, 1, 2, 1, 2, 7, here 1, 2, 7, right. And to a acyl carrier protein is attached 4-phosphopentethene and sulfitrile group, right. In the same manner here ICP is there and 4-phosphopentethene is attached and sulfitrile group is attached. One monomer, this is subdivision, one subunit, one subunit. And this division is functional division, right. You want to do it physically? Physically करना है इसको, feel करना है. 7 boys आ जाओ पहले, फिर 7 girls को बुलाएंगे. Both hands, right. The second is at acetyl transacylase है ना? Yes, show them. Third is आ जाओ. Third is our Mother Teresa Hospital, malonyl transacylase, right. Kabir Singh at Mother Teresa Hospital, New Delhi. The 4 enzymes are clear to all of you. Then what he got for relief? In all reductase for relief, right. Then ketosyl reductase हो गया. कीटोसाइल किसके पास है? और इसके साथ क्या लगा था हमारा? एक गुड़ी आ जाए, एक गर्ल आओ जल्दी, और उसका काम. So next is acyl carrier protein and it is having 4-phospho, एक ऊपर एक नीचे. See the diagram. Acyl carrier protein is there, 4-phosphopentethene is there and sulfityle group is attached. Clear? Next comes thioesterase. कहाँ गया हमारा thioesterase? Yes, this is our thioesterase. Clear to all of you? I started from the first enzyme, that is our ketosyl synthetase. Show them clearly. Acetyl transacylase, malonyl transacylase. Then fourth is dehydrogenase, fifth is enol reductase, then ketosyl reductase. Then comes acyl carrier protein. See the diagram. Anushka, I am showing you alag. And it is attached to 4-phosphopentethene and sulfitrile group. Next, the 7th enzyme is thioesterase. Clear to all of you? What was the mnemonic? Kabir Singh. Kabir Singh was uneasy due to acidity. He was residing at Delhi, so he went to Mother Teresa Hospital. Clear? Then he got ENO and he got relief, right? Then the ENO got stolen from the pharmacy shop. For further more packets, he went to pharmacy shop, but keto was it was stolen by cats. It was came in the CCTV footage. Keto, kitty, ketosyl reductase, right? Then cats was handed over to our ACP of police, and ACP take it to the Thane thioesterase. Clear? And this is one monomer. अब 7 गर्ल्स और आ जाओ. Now I told that it is a dimer and dimer is functionally active, but it is antagonistic. Now the first enzyme will come at this side. Come, my daughter. So there was, first enzyme was Kabir Singh. Where is it? Kabir Singh, show Kabir Singh. Yes. Now here it is antagonistic. Second enzyme is acetyl, show them, acetyl translylase. Third is our Mother Teresa Hospital, and he went to, I lost one of my charts. So this is our dehydrogenase, right? Kabir Singh was there at Mother Teresa Hospital, New Delhi, right? Then he got relief via enol reductase, and that eno was stolen by kitties, right? It came into the CCT footage, and then this ketosyl reductase was caught by ACP, right? ACP took it to the Thane, and this is ACP is attached to 4-phosphopentadiene and sulfate group, right? So these are diagonally going on. I started from first enzyme here, and remember one thing, unity strength, the dimer is functionally active. Monomer will not work only, right? So the first enzyme is our ketosyl synthetase, acetyl transacylase. Tell me the name, speak. Malonyl transacylase, dehydrogenase, enol reductase, and this is our right ketosyl reductase. And this was caught by ACP, then 4-phosphopentadiene, and the last enzyme is our thioesterase, right? Now the second chain of dimer is in antagonistic way. So I start from the first enzyme again. The first one was there, now the first one will come here. What is the enzyme? Kabir Singh ketosyl synthetase. Now the second is Kabir Singh was at acetyl transacylase. Then Mother Teresa Hospital gaya. What was the name? Malonyl transacylase. Then Delhi dehydrogenase got relief via eno, and got the eno was stolen by kitties, ketosyl reductase, and this attached to, it was caught by ACP of police, acyl carrier protein, and finally handed over to police station Thane. So hope you remember this. It is very easy now, Hana? What was the mnemonic? Anybody who can recall and help me revise? Mr. Singh, you can try. Singh, let's try. Yes. Tell me the first enzyme. Ketosyl was Kabir Singh. The second is enol reductase. Acetyl transacylase. Then he went to Mother Teresa Hospital, malonyl transacylase. The hospital is situated at New Delhi, dehydrogenase. Then relief via eno, relief enol reductase. Then the eno packets were stolen by cats. The cats are ketosyl reductase, right? And then this cat was pakda by who caught it? ACP, and ACP handed over to Thane, thioesterase, right? The one monomer is clear to all of you. Now this monomer is antagonistic. Starting from first enzyme, this was Kabir Singh, ketosyl reductase, then at acetyl transacylase, then Mother Teresa Hospital, malonyl transacylase. Handed over to this hospital is situated at New Delhi, dehydrogenase. Then enol reductase, eno, and relief. Then this eno was stolen by kitties, ketosyl reductase, handed over to ACP of police, and police handed over to lockup Thane, thioesterase, and this ACP is attached to 4-phosphopentadiene and sulfate group. Now it is very easy, I think so, right? So fatty acid synthesis complex is okay. Okay, any doubts? Ask. Yes, ask. किसी को डाउट है तो बोलो, नहीं तो फिर मैं पूछता हूँ एक बार. बोलो. वो हंस रही है बहुत ज़्यादा, बहुत हंसी आ रही है. आजा बेटा मेरे साथ ही आजा, बैठते हैं, इकट्ठे पढ़ते हैं, लेक्चर लेते हैं, आजा. बोलो, डाउट है तो बोलो. It 100% 5 marks का क्वेश्चन आएगा. He will ask for diagram and the enzymes. Now you can remember this by this monomer. Kabir Singh at Mother Teresa Hospital, Delhi, right? He got a leaf fire in all ductase, ENO. This ENO was stolen by kitties, cats, cytosol ductase. And this cat was caught by ACP, acyl carrier protein, and then ACP handed over to the police station lockup, Thane, thioesterase, right? One monomer is clear, and then again antagonistic, right? The dimer is, they will form strong bonds, right? The dimer is functionally active only. The monomer will not work all alone, right? And next of the features, this is color diagram. Ketosol synthetase, acetyl transylase, mannolite transacylase. Then dehydrogenase, enol reductase, ketosol reductase, thioesterase, and acyl carrier protein. And acyl carrier protein for phosphopentethine and sulfitryl group. In the same way, we start in antagonistic manner. Now clear to all of you, we have done it physically. It is a multi-enzyme complex, right? 7 enzymes are there and 1 ACP is there. Who caught them, right? And dimer with 2 monomers. 2 monomers are there and dimer is functionally active, right? And all the enzymes originated from a single gene. And there is good coordination between all the enzymes, right? And other metabolites, they do not interfere in this complex, right? And dissociation of this complex result in loss of, agar ye complex टूटेगा, enzyme activity will be lost. And 2 identical subunits, the molecular weight is 2,40,000. Viva question, multiple choice question, the molecular weight of 2 identical units is 2,40,000. 7 enzymes are there and 1 ACP is there attached to 4-phosphopentethine and sulfitryl group, right? You can remember in the 3D. Next, the 2 subunits are anti-parallel, right? Ketosol is here. Raise your hand, ketosol. Ketosol, number 1. Kushmita, raise your hand, right? Diagonally. Second enzyme, raise your hand there. Second. कौन है दूसरे नंबर पे? कुश्मिता के साथ वाली, right? Third raise. Fourth, dehydrogenase. Then this, okay? Now next. So we have had to tail and tail to head orientation of the enzymes, right? And the dimer is functionally active. Unity is strength, the dimer will work, otherwise monomer will not work. And they are complementary, I told, 1 to 7 and 7 to 1, right? And it is very great efficient complex and there is no permeability barrier. There is no permeability barrier is required and there is good coordination between all these enzymes, right? So the diagram from Satyanarayana is there. I hope you can now remember. अगर घर जाके 5 minutes भी लगा लोगे तो it will be easy. क्योंकि अब हमने इसके आगे reactions करने इसके सारे enzymes के, तो you have to learn this first. My promise 5 marks question आएगा इसमें से final में. अगर FA में डालेंगे तो भी आएगा, उसमें भी आएगा. Clear? Any doubts? So you can leave. Thank you to all of you for supporting me. Yes. This, right? It takes place in which organ? Liver, kidney, adipose tissue, and lactating mammary gland. Right? And the raw materials are acetyl CoA and NADPH, and the third is ATP. Acetyl CoA is formed in the matrix, but fatty synthesis takes place in cytosol. So acetyl CoA has to be brought to the cytosol, right? When acetyl CoA is in the cytosol, it will require NADPH and ATP will start with fatty acid synthesis. The first step is conversion of acetyl CoA into malonyl CoA, right? From malonyl CoA will enter proper fatty acid synthetase complex where all the reactions take place, right? And fatty acid synthetase complex has how many enzymes? 7 enzymes and 1 acyl carrier protein attached to sulfitryl group via 4-phosphopentethine, right? The properties are discussed, so that is over. In next class we will discuss the reactions of fatty acid synthase complex. Thank you.